The overall goal of this procedure is to visualize migrating neurons in living brain slices. This is accomplished by first dissecting the cerebellum from a P10 rat. The next steps are to prepare acute cerebellar slices and to label the neurons with a fluorescent probe. Then, the time-lapse experiment is performed through macroconfocal imaging or confocal macroscopy. The final step is to track the neurons in the resulting movies. Ultimately, ex vivo macroscopy is used to show the role of endogenous factors or toxic substances that regulate neuronal migration.
The main advantage of this technique over existing methods, like confocal macroscopy, is that macroconfocal imaging increases the field of view of living cerebellar slices directly placed on a membrane insert.
I first had the idea for this method when I had difficulties to transfer and stabilize cerebellar slices for confocal microscopy experiments.
Begin by having accessed the skull of a decapitated P10 rat puphead. Using fine iris scissors, delicately make 2 lateral incisions from the base to the rostral region of the skull. Then, remove the dissected skull with 2 No. 3 forceps, breaking any adhesion between the brain and the skull. Now, scoop the brain out with the spoon end of a spatula and put it into a 35mm Petri dish with 2 ml of cold HBSS